All right, so we'll get right into it. Our first speaker is Dr. Manoj Lalu from Ottawa.
Okay, great. Thanks so much for the opportunity to give this lightning talk. This is on behalf of my co-leads of the National Preclinical Sepsis Platform, or the NPSP. It's Dr. Braden McDonald and Dr. Asher Mendelson, as well as on behalf of all the NPSP investigators, several of whom are here, and I'll mention them as well. So what we've been really interested over the last couple of years is taking a multi-center approach to laboratory work. As many of you are aware, multi-center clinical trials are a gold standard approach to conducting clinical research. And we think taking that same approach might hold advantages in the laboratory as well. So for instance, it could help promote collaboration. It could help promote information exchange, refinements in methods and research practices as well. And of course, this sort of approach can help immediately assess generalizability of findings. So if my lab finds something, and Dr. Sean Gill's lab, who's one of the NPSP investigators, finds the same thing, Dr. Patricia Liao's lab finds similar findings as well too. Well, number one, it demonstrates that that finding might be generalizable across multiple laboratories, but it also speaks to the fact that perhaps we're closer to the ground truth of whatever we're looking at as well. So over the last few years, we've been meeting quite regularly, almost on a monthly basis at some periods. And we're truly pan-Canadian with multiple participating centers, along with those labs that are listed here in the map. We also have centers in Dalhousie, led by Dr. Lehman, who's here in the crowd as well too, and Montreal, JS Jael. And we've basically been meeting, as I mentioned, to develop protocols and also develop a number of ancillary projects that I'll mention at the end here as well too. One thing that we're very proud of is that these meetings are not just attended by PIs. They're also attended by almost all of our HQP, including Michaela, who's the host here today, as well as veterinarians, so our animal veterinarians from the different centers. And over the last year, we've also had patient partners, I think some of whom are presenting here as well today, but I don't see any in the crowd right now. So it's been a great sort of gathering of minds. And oh, sorry, I should mention also that right after our inaugural meeting, we actually published a proceedings paper where we talked about our general approach and what we plan to do. So that's that paper on the bottom there. So we're allowed to present one piece of data, and I thought I would give you a quick overview of our first paper that came out just a couple of months ago, where we've characterized a 72-hour model of intraperitoneal sepsis, and this is called a fecal-induced peritonitis model, or a FIP, so that's the FIP on the top there. What you can see is a mouse here that we basically inject with fecal slurry, and we follow them for over 72 hours. And during this period, we basically give them painkillers, so buprenorphine, and we also resuscitate them with fluid and imipenem. Those are the different arrows you see across the experimental time course. So looking at two different labs, my lab and Dr. Patricia Liao's lab, you can see that survival of these animals was, it was relatively similar. There's some differences there. So one lab is in blue, another lab is in green, and then the black line are just healthy animals. None of them had any problems health-wise over the time course. What's really interesting, though, more so than that survival curve, is this piece of data where we have a clinical score that we use. It's called the Mouse Sepsis Score, or MSS. This is just findings from one laboratory, but we did see very similar findings in the other laboratory. And one thing you might be not aware of is the fact that we can't let animals die of sepsis. So for humane reasons, we actually have to monitor them as they're sick. And when they reach a critical endpoint or humane endpoint where an MSS score has reached a threshold, we actually sacrifice them. So as you can see, some animals actually reach that threshold and we sacrifice them. However, what was very surprising is that some animals never reached that threshold and they were unfortunately found dead. And this is a concerning finding because the MSS is used internationally by many labs as a surrogate humane endpoint of death. And what these findings suggest is that perhaps we should not be using it for that purpose. Perhaps it is not the best surrogate humane endpoint for death. Finally, I wanted to just mention that we are truly using the NPSP as a platform beyond mouse experiments as well too. So we're actually studying preclinical collaboration. This is a study that's led by Dr. Jenna Evans. She's a social scientist here at McMaster University, where she and her team are studying us as scientists and how we actually collaborate with each other, and also providing suggestions on how we might improve that collaboration. That work was funded, or is funded, by the NFRF. Systematic reviews are very common in the clinical research world. You may not be aware that they're very uncommon in the preclinical research world, but the NPSP is helping change that. We've been funded by the CIHR to produce basically a number of systematic reviews, a couple of which have already been published. And then finally, patient engagement. You'll hear more about that later today, of course, but patient engagement is becoming more common in clinical research. It is very uncommon in preclinical research. But despite that, we've actually been able to bring patient partners onto our team. They've been attending our meetings, as I mentioned. And what we've been doing over the summer is a separate project where we're co-developing a patient-important research question where we ultimately want to answer that question in a lab study downstream. And that's it. So what we're going to do next is conduct the world's first sepsis multi-laboratory study, looking at fluids and antibiotics and the intersection with biological sex. There is a poster out in the hallway at noon where Dr. Farouk Jahandade, you can just raise your hand there, Farouk, she'll be presenting that poster. I encourage you to take a look at that, and you can see some of our latest results around this 8-hour model. I'll just leave this up here while we take a minute of questions, I guess. Thanks, everyone.
Okay, do we have any questions for Dr. Lalu?
Alex's question. Thanks, Manoj. So what's the difference between the animals that were humanely killed and the ones that were died? Did you do any autopsy or
no, we didn't. And that's one question, actually, Alison was mentioning yesterday. You know, we got to find out what they're dying from, basically, right? So it's almost certainly some form of multi-organ failure, but we have to take a deeper dive into that. So it's a good question.
Hey, thank you so much. I was wondering if there was a specific interest in specific outcomes, either short or long-term in mice, and if you had considered looking at the pathway from sepsis to cardiovascular disease, which is one of my interests, and I'm biased.
Yeah, no, it's an interesting question. It's something that's actually been brought up from that patient engagement program that I talked about at the very end there. Certainly, sepsis survivorship, which has been brought up even yesterday multiple times, was brought up, and in those discussions with patient partners and the researchers. So that's certainly something that we're going to look at. The issue with long-term studies is they're very expensive in mice. So we have to pay for every day that they're in the housing facility, which adds up once you start doing lots of different mice. So yes, we want to look at that, and whether it's cardiovascular disease or other aspects of survivorship afterwards.
Just to add to Fetty, one of my grad students actually has a poster about a protocol in terms of just actually doing that, Fetty. So I've been thinking about that for a while, but Manoj is right, it gets really expensive. And what the model is difficult as well.